Well, because we've got to overcome, remember, if we're going gluconeogenesis, we've got this irreversible step just before pyruvate. So we got to go around that irreversible step.
You're just doing it inside the mitochondria.
Right. Part of it is going on inside the mitochondria. Any questions about that?
Can you explain something about transporting to electrons?
Well, right, when NADH, right, we said the NAD coenzymes receive when they participate in oxidation reduction reactions, they receive a hydride ion. Right? The hydride ion is this. 2 electrons. And so when they give up that hydrogen, they're giving 2 electrons to this to make this. Oxaloacetate, let's see if I've got it. Well, here we go. Oxaloacetate is a ketone. I don't have the structure here. Malate is an alcohol there. So when we go from there to there, we're reducing that ketone to an alcohol. It's receiving those 2 electrons. So malate carries 2 more electrons than oxaloacetate does. Right? So malate's got those 2 electrons. It moves out here. It immediately gives those 2 electrons back to NAD to make NADH. That's the 2 electrons we're carrying out.
You said it's pushing it towards gluconeogenesis?
Right, because the malate to oxaloacetate reaction shifts NAD to NADH. If we have high NADH in this reversible reaction, it's going to tend to push this equilibrium more towards that direction, which is going to push these equilibrium reactions in the middle toward gluconeogenesis. Okay?
What conditions would the aspartate pathway be better for?
Oh, that's a good question. Under what conditions would the aspartate pathway be favored? I should know this. I'll have to think about it.
Okay.
Does it do the same thing with photons?
No. No, there's no, that's the difference. There's no, right? We're transferring amino groups out of the mitochondria, but not the reducing equivalents.
And what did you say it's glutamine?
This is
Or glutamic acid?
glutamic acid,
Glutamic acid.
the alpha-ketoglutarate. So this is the amino acid. It gives up its amino group, makes the carbonyl. We'll talk about this reaction specifically when we talk about, pardon me, amino acid metabolism more. Okay? So, we go back here. Right, there's the malate oxalate. That's what that's talking about. The 2 different ways of showing. That's just a different way of showing the 2 ways of shuttling out the oxaloacetate equivalent. The top is the what we were talking about. Right? Oxaloacetate to malate, malate back to oxaloacetate. The bottom one is the amino acid route. We talked about route 2 first. Okay, so now we've got our oxaloacetate. Now we want to make out of that phosphoenolpyruvate. Now, I think this is a little misleading. Let's see if I've got a better mechanism here. I do. Okay. So, for phosphoenolpyruvate, if we're just thinking about the basics of the mechanism, this is another one I have. This is phosphoenolpyruvate carboxykinase, called PEP carboxykinase. It is a 74 kilodalton monomer. There are versions found in both the cytoplasm and the mitochondrion. Distribution varies by species. Humans have approximately equal activities in each compartment. but when we think gluconeogenesis, we consider that most of this is going on in the cytoplasm, not in the mitochondria. So we've got oxaloacetate. This is one that is using GTP, right, as the nucleoside triphosphate. We get GDP, carbon dioxide, and our high energy compound, phosphoenol, right, enol, there I got my en and my ol right, phosphoenolpyruvate. Okay? So, we're breaking the mechanism down a little more than they do in this figure, right? If we carboxylated this, but things where we have a carboxyl group on the carbon alpha to a carbonyl, those kinds of compounds are susceptible to spontaneous decarboxylation because there's a stable carbanion that could be formed right there. We ran into this, I believe, in organic chemistry when we talked about things like the acetoacetic ester synthesis, or the malonic ester synthesis, where we made something and then it spontaneously decarboxylated. If you go back to those reactions in organic, we encountered that situation where carboxyl groups on carbons adjacent to carbonyls become labile, and especially if the enzyme helps. So, the enzyme helps stabilize this decarboxylation. If a pair of electrons from this oxygen comes here to remake the double bond of carbon dioxide, this pair of electrons is free to go here because we end up with a carbanion stabilized by resonance. This should really be a resonance arrow, not an equilibrium arrow. Stabilized by resonance, right? You get that stabilization. So you get that pyruvate carbanion stabilized by resonance to the pyruvate enolate. But this carbanion form, right, the less stable of the two forms, this carbanion form, I keep standing in Kyle's way, this carbanion form is a potent nucleophile, right? I'm sorry. In this case, we're going to direct the enolate form to be the nucleophile. Normally, we would look at the carbanion form, but the enzyme directs the enolate form as the nucleophile. So enolate attacks the gamma phosphate of GTP. GDP can act as the leaving group in a nucleophilic acyl substitution reaction. You get phosphoenolpyruvate plus GDP. That's the PEP carboxykinase reaction. So, interestingly, the whole purpose of putting this carbon on here in the mitochondria is that it can spontaneously come off when we need it to make a potent anion nucleophile. Where we go? Our potent anion nucleophile. So we put the carbon dioxide on pyruvate to make oxaloacetate, not because we needed it there in the end, but because it created an advantageous situation to do the subsequent chemistry to make phosphoenolpyruvate. Okay? Any questions about that? We're going a little faster than we did Friday. So, PEP carboxykinase is hormonally controlled. And this is one of the cool things about PEP carboxykinase. I think this actually appears in like chapter 25 or something, the control chapter, not the gluconeogenesis chapter, but I like to cover it here. And it is responsive to both glucagon and insulin, right? Most gluconeogenesis, right, except for MAPA gluconeogenesis. All tissues that can do glycolysis can do a little bit of gluconeogenesis. But if we're talking about gluconeogenesis to create large amounts of sugar to maintain blood sugar, we're talking about gluconeogenesis going on in the liver. And as we've said before, when we say it's going on in the liver, we also mean it's going on in
Kidney.
the kidney, right? The liver's little brother, the kidney, right? That likes to tag along and try to do things. But quantitatively, the liver is much more important. So do we want to do glucagon or insulin first? Tyler, your choice.
Let's do glucagon.
Glucagon first, because we've done glucagon before. So, glucagon binds to its receptor, right? Like we said before. G protein associated receptor, GTP replaces GDP, the GTP bound alpha subunit dissociates from the complex, diffuses within the inner leaflet to interact with adenylate cyclase. Adenylate cyclase takes ATP and makes cyclic AMP. The cyclic AMP binds to the regulatory subunits, which release the catalytic subunits of the cyclic AMP-dependent protein kinase. We've talked about other targets of the kinase. Now we're going to talk about a new target protein. Our new target protein is this one, CREB, which stands for cyclic AMP response element binding protein. So cyclic AMP response element binding protein. Cyclic AMP response element binding protein. The phosphorylated cyclic AMP response element binding protein diffuses through the pores in the nuclear envelope and can interact with the cyclic AMP response element, which is a signal stretch of DNA immediately before the PEP-CK gene. And that's PEP-CK. I don't know what happened to my K there. Something weird happened. That's a poorly drawn K, right? And this response element binding protein binding to the cyclic AMP response element is a promoter of transcription of this gene. So lots of glucagon means lots of PEP-CK, which means an increased capacity for gluconeogenesis. Right?
It promotes transcription of the PEP-CK gene.
The PEP-CK gene, which means then we make more PEP-CK, which means we increase our capacity for gluconeogenesis. So, I'll do my so later. Any questions about that? Kevin's thinking about it. Are we good?
Well, I mean, I'm guessing there's like a control way or something to like get rid of PEP-CK, where it goes away or something if you have to make it every
Well, every protein has a lifetime, right? A half-life. Eventually, proteins get damaged or they become detected as having been around for a while. The cell attaches a small protein called ubiquitin. And the longer a protein is around, the more ubiquitin gets put on it. And the longer that ubiquitin tail gets, the more likely it is that that protein will be recycled. So there are ways that cells determine the half-life of proteins. So, but half-life is a function of how quickly is a protein degraded and removed from the system versus how quickly is it put into the system. And so those two reach an equilibrium. So if you suddenly increase the synthesis rate, then you increase the amount of protein around at any instant. Does that make sense? And so you increase the capacity. If that glucagon signal goes away, Right, eventually over time.